Which of the given statements is incorrect? A. Before using a sequence file in a sequence analysis program, it is important to ensure that computer sequence files contain only sequence characters and not special characters used by text editors. B. Computer sequence files might contain special characters used by text editors. C. Editing a sequence file with a word processor can introduce such changes if one is not careful to work only with text or so-called ASCII files. D. Most text editors normally create text files that include control characters in addition to standard ASCII characters. The correct answer is computer sequence files might contain special characters used by text editors. Which of the given statements is incorrect about ASCII and hexadecimal? A. Computers store sequence information as simple rows of sequence characters called strings, which are similar to the sequences shown on the computer terminal. B. Each character is stored in binary code in the smallest unit of memory called a byte. C. Each character is stored in binary code in the smallest unit of memory called a bit. D. By convention, many of these combinations have a specific definition called their ASCII equivalent. The correct answer is each character is stored in binary code in the smallest unit of memory called a byte. Which of the following is wrong about genetics computer group sequence format? A. Earlier versions of the genetics computer group GCG programs require a unique sequence format and include programs that convert other sequence formats into GCG format. B. Information about the sequence in the GenBank entry is not included but the line information is carried out. C. If one or more sequence characters become changed through error, a program reading the sequence will be able to determine that the change has occurred because the checksum value in the sequence entry will no longer be correct. D. Lines of information are terminated by two periods which mark the end of information and the start of the sequence on the next line. The correct answer is information about the sequence in the GenBank entry is not included but the line information is carried out. Which of the following is wrong about European Molecular Biology Laboratory Data Library Format? A. EMBL maintains DNA and protein sequence databases. B. As with GenBank entries, a large amount of information describing each sequence entry is given. C. Sequence entry includes literature references and information about the function of the sequence but not locations of RNAs and coding regions. D. Information is organized into fields each with an identifier shown as the first text on each line. The correct answer is sequence entry includes literature references and information about the function of the sequence but not locations of RNAs and coding regions. According to standard amino acid code letters, which of the given pair is not right? A. K lysine. B. Y tyrosine. C. Q glutamine. D. R serine. The correct answer is R serine. Which of the given statements is untrue? A. Sequence and other data files that contain non-ASCII characters also may not be transferred correctly from one machine to another and may cause unpredictable behavior of the communications software. B. The ASCII mode is useful for transferring text files and the binary mode is useful for transferring compressed data files which also contain non-ASCII characters. C. ASCII and binary modes cannot be set by the user. D. Most sequence analysis programs also require not only that a DNA or protein sequence file be a standard ASCII file but also that the file be in a particular format such as the FASTA format. The correct answer is the ASCII mode is useful for transferring text files and the binary mode is useful for transferring compressed data files which also contain non-ASCII characters. Which of the following is wrong about National Biomedical Research Foundation Protein Information Resource Sequence Format? A. Sequences retrieved from the peer database are not in this compact format but in an expanded format with much more information about the sequence. B. The NBRF format is similar to the FASTA sequence format but with significant differences. C. This is different than peer format. D. The first line includes an initial character followed by a two-letter code such as P for complete sequence or F for fragment followed by a 1 or 2 to indicate type of sequence, then a semicolon, then a 4 to 6 character unique name for the entry. The correct answer is: this is different than peer format. Which of the following is wrong about FASTA sequence format? The FASTA sequence format includes a comment line identified by a character in the first column followed by the name and origin of the sequence. B. The FASTA sequence format includes the sequence in standard one-letter symbols. C. This format provides a very convenient way to copy just the sequence part from one window to another because there are no numbers or other non-sequence characters within the sequence. D. The presence of is not quite essential for reading the sequence correctly by some sequence analysis programs. The correct answer is: the presence of is not quite essential for reading the sequence correctly by some sequence analysis programs. Which of the following is wrong about abstract syntax notation sequence format? The information is much more difficult to read by eye than a GenBank formatted sequence. B. Abstract syntax notation ASN.1 is a formal data description language that has been developed by the computer industry. C. All the information found in other forms of sequence storage, e.g. the GenBank format, is present. For example, sequences can be retrieved in this format by ENTREZ. D. Taxonomic information and bibliographic information cannot be encoded with this format. The correct answer is: taxonomic information and bibliographic information cannot be encoded with this format. Which of the following is wrong about GenBank DNA Sequence Entry? The information is organized into fields, each with an identifier shown as the first text on each line. B. In some entries these identifiers may be abbreviated to two letters, e.g. RF for reference. C. Some identifiers may have additional subfields. D. The CDS subfield in the field features does not offer the amino acid sequence. The correct answer is: the CDS subfield in the field features does not offer the amino acid sequence. A consecutive set of three-letter words that could be codons specifying the amino acid sequence of a protein. The sequence entry is assumed by computer programs to lie between the identifier's origin and A. True, B. False, C. D. The correct answer is true. For computer analysis of proteins, it is more convenient to use single-letter than three-letter amino acid codes. A. True, B. False, C. D. The correct answer is true. In organization of the GenBank database and the search procedure used by ENTREZ, each row is another sequence entry and each column another GenBank field. A. True, B. False, C. D. The correct answer is true. In Stanford University Intelligenetics Sequence Format, at the end of the sequence a 1 is placed if the sequence is linear and a 2 if the sequence is circular. A. True, B. False, C. D. The correct answer is true. The format of an entry in the SwissProt protein sequence database is very similar to the EMBL format. A. True, B. False, C. D. The correct answer is true.